So actually when I started in late 90s or mid 90s, so I started as an interest. That time antimicrobial resistance things were not that common. The bacterial system is a very nice model system to understand gene expression, molecular basis of gene expression. So my interest started there. Then as my career progressed, the AMR thing came into the picture, and then one can start thinking of how to apply your basic science knowledge to understand this biotechnological problem or pharmaceutical problem. So in that way, my knowledge of research also could be used to understand antimicrobial resistance. See, the bacteriophage, as I was discussing during the talk, that it has a lot of machineries which can manipulate the bacterial DNA. Now what happened, as I was telling, that you can use it for your own purposes. One of them is CRISPR-Cas9. It's a beautiful system to edit, change mutation, put a DNA fragment in. And so people took it for genetic engineering and gene therapy in that sense. Now phage therapy is a little different. The basic common part is that phage therapy has 2-3 different aspects. One is using the bacteriophage itself to kill the bacteria. So this is in use for a long time. So like before antibiotics were discovered. The problem is bacteriophage, you cannot just drink it or inject it in the bacteria in the blood, because a lot of immune response will come and there's a problematic. So other way, so people are now trying to change the bacteriophage itself, something called humanization of the bacteriophages. So that's another aspect. Now phage therapy is that aspect. Second aspect of phage therapy is that you can use phage particles as a vehicle to inject something. That's the one aspect. Third aspect is what I was talking about, that you use phage proteins for certain biotechnological use. Just the CRISPR-Cas9 is one of them. So CRISPR-Cas9 is one aspect. There could be other molecular biological tools that in most of the laboratories we use. We cut and paste DNA restriction enzyme, then ligases are there. As I gave an example of in vitro synthesis of RNA, RNA vaccine, a lot of T7 RNA polymers, as I gave an example. So that is another aspect of phage therapy. So you are thinking of taking of molecules from phage, which are very interesting. So they are already there. Nature has already made it to manipulate bacterial genome, so you can use it for your own purposes. So like we were looking for certain proteins which is phage coded, but at the end what you were getting a lot of synthetic things which are unnatural things which probably will not able to design by ourselves, but in the process of genetic selection we could able to actually make those synthetic proteins. So I feel like those are very exciting part. Well, that they can become a part of a breakthrough research or not, that is a time will tell. But as a researcher, these are the exciting moments, right? So that makes you to stay as a researcher. One aspect is to understand the basic molecular biology processes. Second is how to use it for different biotechnological uses. So in that, and important thing is that you don't really need a lot of very expensive infrastructure, right? For university setup or for a new department like biology department, I think this is more important field. And I find faculties, at least 5-6 people that I interacted, they are pretty enthusiastic, and it looks like they will be succeeding, and hopefully university will be supporting more. And the most important thing is doesn't require a lot of infrastructure. Mostly it is intellectual driven. If you have a nice thought process, you can actually succeed. I think should be complementary to start with, because still the bacteriophage therapy is a long way. It is, as I told you, that you just cannot use the bacteriophages you cannot inject it to the blood or something. So that is one big challenge. But yes, it is a very promising field. There are many bacterial infections, especially those which are on the surface. There is a famous case of cystic fibrosis in the lung, so which was totally incurable. I mean, it was resistant to all the drugs. So the phage cocktail helped work with. So I feel that it will be complementary in the future. But the biggest challenge is you have to use it, humanize it. I mean, it should not have a very strong antibody response or immune response. So then only phage therapy can come into the picture.